the focus of our project is developing stapled peptide PROTACs, which is a new technology platform where we try to deliver the punch of 3 drugs in one. And the goal is to try to activate and deactivate the relevant pathways in cells so that we can get cancer cells to essentially disappear. And we are very focused on particular types of cancer cells that retain an intact p53 pathway, p53 being one of the most important tumor suppressors in biology. And those cancers that retain p53 are uniquely susceptible to our approach. And in 90% of pediatric cancers, they retain this pathway, and roughly 50 to 60% of adult cancers retain the pathway as well. So we're hoping for a broad impact of the approach. When I spoke with you last, we had started this project to try to show that it could actually work across a spectrum of cancers. We were trying to figure out which would be the types of cancers that would be most susceptible to our treatment. And then we went ahead and looked in different cancer cell lines to validate those hypotheses. And we're heading toward identifying what our lead compound would be. In the second year, we were able to successfully identify a lead compound that had the best activity from anything that we had made before, and we very much focused with our stage 2 funding on trying to push this all the way through analysis of stability, pharmacology, and then in vivo testing. So literally in the last 2.5 months, we did our first in vivo efficacy test, in other words, testing our compound in a mouse model of cancer. And based upon the pharmacology that we had done, we were able to identify a route and a dose that we predicted would be the most effective. And we just completed our first animal study, and the results were beyond anything that we had expected. We were actually doing a pilot study, and in the pilot study we saw dramatic shrinkage of the tumors, and we started with tumors of a pretty high burden. And our animals that were treated with vehicle, they lost weight, the tumors continued to grow, and we were able to actually prevent weight loss, and we were able to shrink tumors. The Harrington Discovery Institute process for these grants has been different than anything I had experienced before in several ways. The first way was that once the funding started, there was seed funding to try to basically see what you could do with the initial tranche of funding to get biochemical results done, to get lead compounds made, figuring out which would be the best one. And I think that really provided an acceleration of this entire project. By the time we applied for the stage 2 funding, the momentum shifted now toward trying to make this matter to treat cancer in a species, in mice, to actually show that you could do something. And so with the second tranche of funds, we immediately deployed it to do experiments that we frankly have never really had the finances to do, which is formal pharmacokinetics, formal stability testing, formal dose finding, before even trying to put this into an animal to treat an animal model. I think the third part of this is that you have a team of seasoned drug developers literally there, standing next to you the whole way, where you can ask them questions, get their advice in real time, and have that influence the way you design and execute experiments. We have done more in 2 years than I ever thought that we've been able to do, and we've only just begun, because now we can look forward to the next round of funding, or a launch of a new company to try to help us bring this forward, or a license to an existing company. And those are the types of things that are there with this entire group helping you achieve it. You're not alone. You're doing it with a team of highly experienced and sophisticated experts to kind of help this become a reality.